In this section, we take a deeper look into the components of the genome and how the different segments we find in the genome came to be. We focus on transposable elements, simple sequence DNA, and multi-gene families.
For most eukaryotes, the protein coding regions of genes and genes coding for rRNAs, tRNAs, and miRNAs only make up a small part of the genome. For us, only about 1.5% of the human genome actually codes for proteins or is transcribed into rRNA or tRNA. The rest of the genome is comprised of regulatory sequences, introns, unique non-coding DNA, and repetitive DNA, both related to transposable elements and unrelated. This figure shows the distribution of the different components of the human genome.
Among the non-coding DNA are fragments known as pseudogenes. Pseudogenes are former genes that have accumulated mutations over a long period of time and no longer produce functional proteins. Although non-coding DNA was once called junk DNA, the high level of conservation among different species suggests some important function that we have not yet discovered.
Transposable genetic elements or transposable elements are stretches of DNA that can move from one location to another within a genome. They are found in both prokaryotes and eukaryotes and move by a process called transposition, similar to recombination. The idea of transposable elements or movable genetic elements was first described by geneticist Barbara McClintock in her breeding experiments with Indian corn. Although at first she was discredited, she eventually won the Nobel Prize after transposable elements were found in bacteria.
Movement of transposons and retrotransposons. The first type of transposable element is transposons, which move by either a cut and paste mechanism, where the elements are removed from the original site, or a copy and paste mechanism, which leaves a copy behind. The enzyme transposase aids the process.
Retrotransposons differ from transposons in that they move by means of an RNA intermediate rather than a DNA intermediate. This entails the synthesis of an RNA version of the retrotransposon and then a DNA version of the reverse transcription completed by the enzyme reverse transcriptase. This achieves the same effect of the copy-paste mechanism mentioned before, necessarily always leaving a copy of the original segment in its original location.
Transposable elements make up about 25 to 50% of most mammalian genomes and are even more prevalent in plant and amphibian genomes. In the human genome, the ALU and line 1 groups of sequences are very prevalent transposable elements, making up about 10 and 17% of the human genome respectively. ALU elements are usually about 300 bases long and code for RNAs that regulate gene expression. L1 retrotransposons are much longer at 6,500 bases long, and they are thought to play a role in the diversity of different neural cell types. L1s have a very low rate of transposition. To reiterate, the transposable elements are included in the non-coding DNA category because their proteins do not usually carry out normal cell functions.
Repetitive DNA that has probably arisen from mistakes during DNA replication or recombination comprises about 14% of the human genome. This can result in long stretches of DNA that are duplicated in different regions of the genome. A different type of repetitive DNA is simple sequence DNA. These structures contain many copies of tandemly repeated short sequences. An example would be the following sequence.
Some units consist of repeats of hundreds of nucleotides, while some contain repeats of fewer than 15 bases. Short tandem repeats, or STRs, are examples of these, with repeats of 2 to 5 bases. The number of repeats varies between individuals and even between two homologs in an individual. These features make STRs helpful in determining genetic profiles that can have applications in paternity testing and forensics. However, this makes it harder when approaching genome sequencing with a whole genome shotgun approach.
Much of the simple sequence DNA is located at telomeres and centromeres on the chromosome, suggesting a possible role of DNA in chromosome structure. Simple sequences prevent gene loss at the tips of chromosomes by protecting the ends from degradation.
Multi-gene families are collections of two or more identical or very similar genes and are separate from the genes present in unique sequences. For families of identical DNA sequences, their final products come in the form of RNA. An example is when the 3 largest rRNA molecules are transcribed from a repeated unit in the genome. The repeated copies of the rRNA allows cells to make the ribosomes used in protein synthesis.
For multi-gene families of non-identical genes, one example is how the 2 related families of genes that encode globins encode either alpha globin or beta globin. Each type is expressed at different times in development. Now, hemoglobin can function in the changing environment, such as aiding younger humans in having higher oxygen affinity.
Analysis of multi-gene families can be used to uncover more information about genomic evolution. The duplication, movement, and divergence of segments of DNA, although most often detrimental, when positive, provide a raw material that drives the evolution and specification that is seen today.
The following are the references that we used to complete the project. Thank you for watching our presentation.